Hi, hello, namaste. This is the Race 3 tutorial, your Srinivas. Today, I am going to discuss a wonderful topic like what is that? Biotechnology and its application, botany, IP question. So, previous videos already I explained principles of inheritance and molecular base of inheritance and plant growth development. Each and every lesson, a very clarity I am explaining botany IP question. So, today, just concentrate, just starting before, please subscribe my channel, like and share it. Let's start my concept. Today's question. List out the beneficial aspects of transgenic plant. First of all, what is a transgenic plant? The transfer of genes from one plant to another plant. Is there any beneficial? There are asked questions. So here the first one is like what is that? Transgenic plants is useful for resistance like what different is given. Just I'm giving you like shortcut code RN hit bars. If you know this code, you can write everything answer. Just here RNHITDAS. This is the code. By the helping of this code, you can write every answer. How sir? Let's see. First one is like what is that? R. R means resistance power. Sir, we are changing from gene from one plant to another plant. So like this is useful to us. Like what is that? A resistance power will get. So best example already we know the BT cotton, BT abbreviation Bacillus thuringiensis cotton resistance to insects are there. Hey, insects, you come and attack, nothing will happen. Next, one question is there, 4 marks question. Very clearly, I will explain to you. So, BT cotton resistance to insects. So, that purpose useful like what is that? Transgenic plant. So, R is the resistant will catch. Next one is what is that? Nutrition value. So, what's the nutrition value? See here, check out nutrition value purpose useful. So it is a transgenic golden rice is often from where types of variety types are. So golden rice means gold color. No, gold color rice are, which may helpful like what is that? Type variety which may produce. What they are useful sir? It is giving vitamin A only. Nowadays we are eating different type of the rice, BBT. There is no only starch we are eating. Complete outside alluron layer is removed out. But by the helping of a transgenic plant variety, newly variety which may produce like golden rice, like that is of typey types are the which may helpful like what is that vitamin A and prevent blindness. So next, so R mean resistance, N mean nutritional value. H is there. H means hybrid seed production. So hybrid seeds are nowadays important in that seedless fruits purpose very useful to us. Like what is that? So third points are there. It is useful for hybrid seed production purpose. Useful. The hybrid seed production purpose what we are using artificial fertilizations are there. At that time emasculation process we are using. So they don't want to need the emasculation methods. Why? Because in that main sterile plants like Brassica, Napas plants are there in that emasculations are eliminated. There is no emasculation. What is meant by emasculation? Removing of anthers from bisexual flowers is called as emasculation. So that's it. That is the third point, Sarji. Next one is what is that? H. In H mean what? It is producing like what is that? Insulin and interferon. Insulin and interferon producing purpose very useful. And also antibodies producing purpose very useful. Next to BA. BA mean what sir? Basmati rice are they. What are basmati rice? Which is helpful like what is that? You know, yes, biryani purpose we are using like basmati rice only. Which may produces like what is that sir here? Biotic, abiotic stress conditions are there. Which is completely controlled. Biotic and abiotic stress conditions are there. And yes mean soya beans are the soya bean in that more proteins is present in this one. Around of ready soya bean, it is herbicide tolerant are there. So I think you are catch by the helping of hard and hit bar, we are writing complete this concept. Come on, take screenshot students. You read it this one only and will take out and write it to your book also or in hit bar. Easily you will read it. Next, the second question I'm going to start. The second question is give a brief account of BT cotton. So cotton crop is a very important economic crop. We want to protect this crop is very, very important. Why? Because this cotton which is infected by what type of insects you know? LCD, Lepidopteran, Coleopteran, Diphtheran. So these three types, LCD, Lepidopteran, Coleopteran, Diphtheran. So that is Lepidopterans like what is that? Tobacco bud worm and army worm. Shortcutly, AT will catch. Coleopteran beetles. Dipterans are FM radio. That's it. So these three types of insects, which is yes completely spawning like cotton plants are. This production purpose, the protection of cotton purpose, we are using newly variety which is produced, that is the BT cotton we are producing. So newly we are created, that is the Bacillus thuringiensis cotton sardi. So these Bacillus thuringiensis which is produced like what is that? Toxic proteins are they. So that's why these toxic proteins are kill like what is that? Lepidopteran, Coleopteran and Diphtheran. So newly modified like BT cotton which is may produce. So that's why what happens regularly how they are coming, same like this they are coming. They don't know we are produced. That what is that? BT cotton which is releases like what is that? Toxic protein and kill the insect like what is that? LCD. So that point is given. Some strains of Bacillus thuringiensis produce toxic protein to kill insects such as LCD, Lepidopteran, Coleopteran, Diphtheran. This is the first point. Are you catching this? The first point is be careful. Lengthy one is there. Second is easy. That BT toxic proteins are startingly what happens sir? Inactive stage is present. Inactive is there. After that what happens? It is active form it will come. And after that what happens? It is slowly and this active form of proteins slowly enter to Lepidopteran, Coleopteran, difference of mid-guard cells and finally it creates pores are there and lysis of cells and finally death the insects. So how sir this BT toxin is coded of genes like insects of genes that is called as what we'll call cry only present. So cry is the different three types are they. What are they sir here? Cry 1AC, 1AC, here 1AC, 2AB. Both are controlled like what is that cotton bollworms are they. Both are same. And cry 1AB, only one AB, one first AB, A, 1A, B, C. So, carnivora, this is the psychological clue you can catch out. First one is A, B, C, that's it. So, very simple, cotton is a protection purpose. Newly we are created BT cotton. How they are produced? Internal toxics are they. So that's why regularly how they are coming, same like this that they are coming, but they don't know, simply they are releasing BT toxin and kill the insects. That's it, very simple, no? Second question is completed. Come on, take screenshot students. And next third question. Let's start third question. What are the some biosafety issues concerned with the genetically modified crops? So we know in previous classes we have studied like what is that transgenic plants. Like a starting question. The transfer of gene from one plant to another plant is useful we are studying. How much of useful is there in the semi harmful also present? So what type of harmful? This is the answer. That is biosafety. This is understanding purpose. I created one code AT and AT. Next one is NGPSN. So NGBSN. ATNGBSN. How, sir? First one is there is a fear of allergens and toxins. So genes we are changing from one plant to another. Sometimes allergies is there, and toxics also produced in human beings and animals. This is the first one. A mean allergy, and another one T mean toxics are there. Toxins also produced. Actually, genes we are changing from one plant to another plant means naturality is not there. Complete natural vegetables is not available. Changing is present. Third one, the risk of the gene pollution. Genes complete pollution is there because of we are changing gene from one plant to another plant are there. So that's what happened. Gene pollution. So natural vegetables not present. What happens, sir? Gene pollution is present. Complete gene polluted. Next, changes of biodiversity. B mean what, sir? Biodiversity. Next one is what happens, sir? Superweeds is increasing. What we are cultivating, that plant is not growing. Another plant is showing growth, are they? That may infect to another plant, are they? So that's what happens, sir. Yes means superweeds. Superweeds are increased. The next is one more. N is the natural evolution pattern which may changes. So this is the code you will catch out. ATNGBSN. At NGBSN, that's it. At NGBSN. Completed answer. Next, fourth question. Next, fourth question is give a brief accounts of pest resistant in plants. So tobacco is one of the economic crops are the this tobacco crops is production purpose. The scientists newly technical method there are produced. This root of tobacco plants are the this is infected by what is that nematode that is Meloidigine incognitia. So this root of tobacco which is infected by one nematode that is Meloidigine incognitia. Day by day, what happens are yield is reduced. So that's why the scientists that are introduced by the helping of biotechnology method, what they are produced, RNAi. RNAi is nothing but what is that, RNA interference. So root of tobacco plant which is infected by what is that? Myeloidigine incognitia. It is one of the nematodes. So how there are protectors are by the helping of RNAi. I think you are catching now up to here. Okay. So this RNAi is involved. Silencing of mRNA is correct. Silencing of mRNA mean what happens, sir? So this Meloidigine incognitia which may infect the root of tobacco plant. So at that time what happens, sir? By the helping of biotechnology method, RNAi is introduced. So that's why what happens, that Meloidigine incognitia regularly how they are coming, like this they are coming. But the root of tobacco plant internal what are they? RNAi is there. So that's why RNAi is entered into nematode plant and that mRNA is silencing is present. mRNA is working is not there means there is no translation process. There is no translation process means there is no proteins. This is the process. So RNAi is how they are produced. So let's will see here. Using by the helping of what is that slowly this is the what happened root of tobacco plants are the so that's the agrobacterium vector and nematode specific gene. Nematode specific gene. Both are introduced within the host cell. So agrobacterium vector and nematode specific gene. Both are introduced into from where? Host plants. Then after what happened? Slowly, which is produced like what is that? Sir? DNA, double stranded DNA are produced. When the agrobacterium vector and nematode specific gene, which is entered within the host organism, which is produced like what is that double stranded DNA. These are DNA, which is produced like what is that sense and antisense RNA. Sense and antisense RNA. So these both are sense and antisense RNA. Both are combined to produce. So here introduction of dsDNA into sense and antisense RNA. So like this process. Then after these two RNAs being complemented to each other to produce like what is that? dsRNAs. These dsRNAs is produced like what is that? RNAi. RNA interference are produced. So this RNAi is introduced within the fruit of tobacco plant. When the nematode genes are entered attacking mean, thus RNAi is entered to nematodes. In that what happens sir, mRNA is not working. So mRNA is not working means there is no translation, there is no protein, finally it may death. That's it. So this is the by the helping of biotechnology method, newly method, which is control himself only, which is indirectly kill the nematode gene. So in this video, 4 questions we have studied. One question guaranteed will come in our coming essay quiz. Please subscribe and like and share it. This is a recipe tutorial.